Very pleasant morning to one and all present here. Please kindly check my voice is audible.
Yes, you're audible, ma'am.
Thank you, ma'am. Thank you for your reply. Uh, we will wait for 5 more minutes. We will wait for that others will join, and then we will start our session, ma'am.
Okay, ma'am. Okay.
Very pleasant morning to all. I'm Premlata. I'm working as an assistant professor from Department of Biotechnology in Sri Sasta Institute of Technology. I had completed BTech Biotechnology in Banariaman Institute of Technology, and then I finished MTech Nanoscience and Technology in Anna University. And I had 2 years of work experience in medical transcription field. And I had presented 5 papers in many conferences, and I had attended many international conferences and seminars while doing postgraduate and undergraduate. So we will start our session. Today we will discuss about genome sequencing and synthetic biology. So yes, kindly check the slide is visible. Slide is visible.
Yes, ma'am, it is visible.
Thank you, ma'am. Thank you for your right. So we will discuss about genome sequencing and synthetic biology today, and we will first discuss about what is meant by genome and how we can sequence the genome and get the data, and how we can store the data in the database, and how the synthetic biology utilize that data so that the synthetic biology will uh, have scope in many field. Using the synthetic biology we can develop many technologies, and many industries are currently working on that, and that is very useful in all the sectors, pharmaceutical, all the sectors. We will see in detail all those things now. So first we will see about the basic functional unit of the cell. Okay, so we will start with the DNA. Yes, we are having cells. We are having cells. In the cell, we are having nucleus. Inside the nucleus, we are having the chromatin reticulum. So while the cells undergoing any cell division or function, the chromatin reticulum get more condensed to form the chromosomes. So we are having 46 chromosomes. Yes, 23 passive chromosome. In the 22 are autosome, and one is the sixth chromosome. So that chromosome only having the genetic information. The genes, if the genes are nothing but the genes are functional unit of DNA. The DNA are tightly condensed in the chromosome only. In case of eukaryotes, the DNA is wrapped around the histone. Okay, histone is the protein part that wrapped around the histone and tightly condensed in the chromosome. Yes. DNA only having the information. Okay, the DNA only having the information. Each and every characteristics of each person are determined by the DNA only. So DNA is only having this ATGC nucleotide base plus nitrogenous base plus. The sequencing is nothing but we are getting this sequence, getting the arrangement of this ATGC nitrogenous base. This arrangement only determines the each and specific characteristics of a person. Okay, and each functional, okay, each every function of the protein, enzymes, we are consuming food. Okay, that food are undergoing digestion, and for the enzyme secretions, all those things, and we are having hereditary diseases. So from the parental, the diseases are carried from one generation to the another generation. All these are due to this sequence only. So while doing that sequence, while getting information, we can do a lot of things. We can eradicate the genetic hereditary diseases. Okay, all those things. So only we need this. So only we need this DNA sequence and the genome sequence. Okay, so we are having approximately 30,000 genes. Okay. And see, each cell is having the gene. Okay, the function, each cell is having the DNA. Gene is the functional part of DNA. So the genes only code for the protein, functional protein. If we are taking the food, we want to maintain the glucose level in the blood so that insulin have to be secreted. So that insulin were secreted by the beta cells of islets of Langerhans in pancreas, right? That cells are having undergoing some molecular biology. Okay, the DNA is undergoing transcription and translation, and then only produce this insulin, and then this insulin act on the glucose so that our blood glucose levels are maintained. So not only this, each every function carried over inside the body. If we need gene, okay, and the gene must produce the functional proteins, okay, and proteins are having playing many roles. Some proteins are acting as an enzyme, some proteins are maintaining the tissue fluidity, okay, some proteins are providing the structure, all those things. Okay, so only we need the particular sequence. Okay, so now we enter into genome. Genome is nothing but the complete set of information. Okay, complete set of genetic information. That is the functional information. If we are having the projects, okay, human genome project that are completed, and we are we are going to extract the complete genetic information of a particular organism. Okay, so that is what genome. So if we have the genome data in our hand, then we can modify the data, we can eradicate the genetic hereditary diseases, or whatever we want to do. If we want to maximize some of the function, if we want to reduce the sum of the function, so when we have that genetic information in our hand, when we have the genome sequence in our hand, then we will do in our required manner. Okay, so see, from the father one part and from the mother one part is only going to give birth a new child. Okay, so this genome, this total functional genetic information only carried from one generation to the another generation. So through this way only the disease or whatever things the the child face are looking like mother, father, all these are due to this carrying information around this nitrogenous basis, that is ATGC. DNA is having the nitrogenous basis, adenine, guanine, cytosine and thymine. RNA is having nitrogenous basis, adenine, guanine, cytosine, uracil. So this arrangement in the complete cell only determines all the characteristics of the child. So if we have that particular sequence in our hand, then we can do. If we want to eradicate some of the diseases in the child, or if we want to treat the disease, we can do. And not only in that way, we will take it out to the further future aspects. Okay, so now we enter into DNA sequencing. Yes, we are having the technologies. Yes, we are having the laboratory technologies, that is we are having Sanger sequencing, pyro sequencing method. In that way, we first extract the DNA and then we will go for the sequencing. Okay, but nowadays the technology has improved. Yes, we are having the computational basis. Okay, we are having gene sequencer. Through that, we get the particular the total arrangement, genetic information, total arrangement of the sequence we will get through the genome sequencer. Okay, and we are having the gene synthesizer. If you want to artificially synthesize gene, we are also having the gene synthesizer. Okay, that are all computationally programmed. How our systems are programmed using the basic algorithm. In that way, we are that whatever things we need to do in the basis of genome information, we will do in the computational ways. Yes, we are having the sequencer with already programmed one. If we give the sample into it, we will get the result. Okay, so likewise, we get the final the genetic information of the whole or the particular organism. So in this way only we are getting the DNA sequencing or genome sequencing. Okay, so now we enter into the synthetic biology. Yes, we are getting the information. We are getting the that ATGC, all the sequences in our hand. Okay, that synthetic biology is nothing but it utilizes all the information. It utilizes the informational technology part. It utilizes the chemistry, basic chemistry. It utilizes the molecular biology, biotechnology, all the fields. Okay, all the fields, basic information, the result of all the fields, and then it utilizes, and then it forms the new way. It synthesizes the new cell. Okay, it is not in the living. It is the non-living. When it synthesizes the new form of cell artificially. Okay, in vitro, in the laboratory, we will do the artificial manner. We will synthesize the cell or whatever we want. We will do it in the laboratory manner by getting the information from the computer science, physics, chemistry, agriculture, microbiology, bioinformatics. Okay, from collecting all the information, this synthetic biology leads to the new way of approaches to treating the diseases or producing the new products. Okay, in every field, in when we take the enzyme production field, or in we take the pharmaceutical vaccines, okay, to diagnose the particular disease, all the field, the synthetic biology is coming as an emerging field. Okay, so now we see about what is meant by synthetic biology. Yes. So synthetic biology is nothing but it will redesign the system. That is, it will gather the information already by doing the laboratory way, and then it redesign the system in our interest. Okay, on the basis of our interest, the if you want to get the product, that is, see the if you want to get the product in high amount, or if you want to produce the antibiotic anyway, if we redesign the pre one, and through that we get the our result. Okay, so for that this synthetic biology are useful, and we can also create a new biological parts, new devices and systems, all those things from the synthetic biology. Yes. Before entering to the core, I present a small video, okay, some video regarding the synthetic biology. If we go through that video, then we will get the deep knowledge on that. Okay, just a second. I will share that video. Once you go through that, you will get the approved knowledge on what is synthetic biology and how it is applicable in all the fields. Uh, Kindly confirm the tab is visible. Kindly confirm the tab is visible.
Yes, ma'am, visible.
Thank you for your input. Just go through this video, get the deep knowledge.
What is it, life? What makes it different from just matter? Everything, living or not, is just chemicals made from atoms. These fish and the water the fish are in are just soups of atoms. But what makes the fish alive and not the water is how the atoms are organized by a special kind of molecule. And you know this because you learned about it in high school, DNA. The double helix molecule that houses the chemical alphabet of A's and C's and T's and G's, which in different combinations can make a flower or a frog or you. Every living creature has its code that makes it grow, reproduce, and change. DNA turns dust into life. And how it does that? Well, now here's the mystery. 20 years ago, scientists learned to read a creature's entire DNA sequence, beginning to end, a genome, it's called. They did this for simple things like bacteria, and then for more complex flies, and eventually for humans. And then they began to tinker, to take the glow from a jellyfish and transfer it to a cat, to make critters do what they'd never done before. And as biologists got better at this, as Stanford professor Drew ND, a new kind of science was born.
It's called synthetic biology. It's a means to an end.
What can we do with the new tools of synthetic biology? Well, we can improve on what's been spelled out over 3 billion years of evolution. We can take it beyond reading genomes or editing genomes and start writing genomes. Our own ideas of what life should be like, making creatures drastically different from any that have ever existed. And how do you do that? Well, already, one group of scientists have rewritten and rebuilt the entire instruction kit for the very humble little yeast. And they plan to put their new recipe into a cell And if everything goes according to plan, it will come alive and begin making baby yeast exactly like its most unusual parent. It will be new in the world. This synthetic yeast will break the continuous chain of evolution that links every creature back to the first living cell. It will be discontinuous, in a way, a thing unto itself. And then where do we take this new technology? How far should we or will we go?
So, yes, we are having the gene sequencer through which we get the gene sequencing, that is AT, that only determines it. Each and the specific characteristics of each organism is determined by a specific sequence. Okay, if you want that sequence to be in the other organism, or if you want to use that particular sequence to get more environmental uses, environmental purpose point of view, all those things we will do whatever manner we will do by the synthetic biology. So this synthetic biology can be done by through approaches, the top-down approaches and bottom-up approaches. That is by dividing this total living system into the simpler manner, or by bringing up by developing the new body for biological parts, and then bringing all the biological parts to do a specific function. Okay, so for if that in that way if we do, then that means that is the bottom up approach. Okay, by doing the synthetically doing the biological parts, and then we organize into a to do a simple function, whatever function we need, then that is the bottom up approach. Okay, so yes, so this DNA sequencing. Okay, this DNA sequencing, after the DNA gets, yes, we are having the databases. The databases is nothing but the where we can store all the data. Okay, yes, we are having the database for DNA sequence. Okay, gene sequence. We are having the database for sequence to already sequenced organisms. Okay, that is simple prokaryotes or single-celled organisms. So that genome are already most of the prokaryotes or simple organisms genomes are sequenced, and then they stored in the database. And we are also having the project called Human Genome Project. Through that also we get the sequence. Okay, that the total human genome sequence that are also stored in the database. Whenever we want for comparison with the other sequence, or we already in the laboratory we had find out the new sequence, and then if you want to compare it with the already stored one, for the all the purpose we compared it with the already stored one. Yes, the National Center for Biotechnology and CBA also having the databases. Okay, what gene database, and then they store that. So by utilizing that, okay, if you want to see, we directly go for application point of view, and then you will get the yes. If we want to produce the enzyme using the yeast, yes, we are having the fermentation technology. In that, what they do, they will grow the microorganisms, and then the microorganisms synthesize the products like ethanol or in amylase, whatever enzymes. So that equally as having the chromosome, right? That chromosome is under that chromosome also having the genetic information. So that chromosome having the sequence, that sequence code for particular amino acid or protein or whatever it able to synthesis. So if we through the synthetic biology, what they do, they synthesize the new chromosome on the basis of already uh, stored one. Okay, they synthesize the new chromosome. That is, they synthesize the new sequence. Okay, that are more energy, that will able to produce the more result. That will give the more result. So what they did, they will create a synthesis a new chromosome and then inserted into the microorganism, and then they undergo fermentation. Through fermentation, we get the final amount in the high average. Okay, so in this way only the synthetic biology are useful. Okay, not only in enzyme production. If we take see when the COVID or any virus entered into the body, our body is producing the antibody against the and COVID virus, right? That is our virus. So through synthetic biology, we can also synthesize a new antibody that is more specific to the particular organism, that is particular antigen. In that way also synthetic biology are very useful, and through synthetic biology we can also synthesize more vaccines, okay, more edible vaccines that are effective in the effective manner. They are all very effective when compared to the commercial vaccines. Okay, they are genetically modified the organism, and they are newly synthesizing the single cell microorganism, so that the pathogenicity of, that is the disease causing effect, will get more effective when compared to the commercial way. So in this way, this synthetic biology are very useful in many fields. Okay, so yes, we are having the gene synthesizer, that is by getting the gene sequence. Okay, we are having the gene synthesizer that is also already programmed one. Through that, we also get the uh, new synthesis, new form of DNA or new form of genetic sequences. After getting that sequence, we are if we promote it to the particular organism, we get the more result. Okay, so likewise, we are also regulate the gene, okay, through the on the basis of circuit manner. We can back regulate the gene or forward regulate the gene on the circuit manner. We are comparing it with the oscillator. We are comparing it with the transistors, okay, voltage regulators. So this basic biological system compared with the electronics way, and then we can use it for the further purposes. See, for example, here is we are this particular sequence for giving the protein that gene need to undergo some of the process. Okay, it should have in the promoter sequence. It should have the particular enzyme binding sequence. All the sequence are we can artificially synthesized. We artificially synthesized through synthetic biology. Okay, that one is called biobrick. Biobricks are nothing but they are all the biological parts. By utilizing the biological parts, what they here what they do, they are taking a particular mechanism from the particular one microorganism, and then another mechanism from the another organism. They cut and then rewind, and then they to synthetically produce the new way of new way of mechanism. Okay, so it contains both our organism mechanisms, so that we get the new one. So with this, we can able to do our required one in the large enormous amount. So for this only we need synthetic biology. Yes, synthetic biology is an emerging field. It is really having many applications, and many industries are working on this in the very in the perspective manner, different manner. Okay. Yes, we are having the another one called pharmacogenomics. That is nothing but if we have the genome sequence of a particular organism in our hand, like if we have the normal person genome sequence in our hand. So if we compare with the another hereditary disease one, we will find out the sequence responsible for that particular disease. Okay, so we can remodulate it, and then we eradicate the hereditary disease in this way. So for that, Okay, for that we use the synthetic biology. Through synthetic biology, we can artificially synthesize the new sequences and then insert into the already one, so that the hereditary diseases are eradicated. So we can able to do um, in that manner. Okay, by utilizing the synthetic biology. So yes, and yes, we are having the genetic library. See here, we are sequencing the DNA and then stored in the genetic libraries. Yes, we are having the genetic library, DNA library, RNA library, protein library. Proteins are nothing but they are on amino acid, right? So through peptide bond, this amino acid are synthesizing the protein. So that protein amino acid sequences are also stored in the library. Protein library, we are having protein database, DNA database, okay, that are called biological databases. So by utilizing the information from that, by through remodeling, redesigning, or by artificially synthesizing the new one, which mimics the already existed one, but have in the more efficient way. So through that we apply for the many field, okay, pharmaceutical field in many fields. Finally, we enter into the companies working on the synthetic biology. Yes, these are the companies: Evonetics, Biomins, Inocular. See, this each companies are working utilize the synthetic biology and producing the many products in the perspective way. Some of the companies are utilizing the synthetic biology and producing the pharmaceuticals, new drug, and some of the industries are working in the electronics basic way. They synthesizing the new promoter and new RNA that is enzyme specific sequences, new tools, automations, all those things. And some of them are industries are working utilizing the synthetic biology for protein engineering. That is for engineer for artificially synthesizing the proteins, so that we need not to depend on natural source for everything. If we are able to synthesize in the artificial manner, if we are able to synthesize the insulin in the laboratory way, if we are able to synthesize anything we want in the laboratory way, enzymes or anything in the laboratory way, then we need not to depend for natural source for all the time. So in this way also synthetic biology are very useful, and it is uh, we are doing all those things based on the synthetic biology. Okay, so in this way only, and some of the industries are working in the food and agriculture. They utilize the synthetic biology for food and agriculture. That is nothing but if we know the sequences responsible for the production of grains, if we know the sequences responsible for the easily getting affected by the any pest or any pest or bacteria or any yeast. Okay. So if we know all those things, then we can remodulate it, or we can synthesize in the laboratory in the de novo, that is new way of synthesis. We are having many manuals, very techniques for all those things. Okay, while by doing that, and then after synthesizing, we insert it into the crop, or we insert it into the organisms, so that we get more grain production, more yield, and also we can make the crop resist to the particular pest or herbicides. Okay, and we also use the synthetic biology for synthesizing the herbicide, pesticides, all those things. Okay. So finally, it is applied in various fields. Okay, in health sector, for biological sensors. We are having the glucose sensor in the handheld glucose sensor, that is within a single drop of blood, we can get the result of blood glucose level within a second, okay, within a minute. So for all those things, we utilize the synthetic biology. Okay, we can produce the more fuels, biofuels. That is, we are having the some of the organisms, okay, oil producing organisms, okay, and through synthetic way we can modulate the chromosomes, whatever genetic information required for producing the fuels. Okay, we can remodulate it. We can do it in the laboratory. We synthesize, artificially synthesized that, and then we go for the enormous production of oil from the particular organism. Okay, and enzymes, and for bioremediation process, okay, we can artificially synthesize the microorganism, which can able to clear out oil spills, or it can able to clear out the uh, oil spills or wastage, that is sewage water treatment. All those things we can utilize the base. We can make a synthetic biology as a basis, and then we can make the new way of microorganisms or new way of chromosomes for all for doing the bioremediation or wastewater treatment on those things. And in the agricultural basis, we will increase our production, okay, production yield. We will increase the uh, fuels. Okay, we will get optimized food production by utilizing this synthetic biology. Likewise, yes, in the future generation, we are going to have biological computers. That is nothing but, see, the more than 3,000 CDs, the storage, the information need to be stored in the more than 3,000 CDs that are we can able to store in a 1 square cube of DNA. Okay, we can store the data in the DNA. So in that way also we can utilize the DNA genome sequencing technology. Okay, we are going to have, as we are having all the research undergoing in that way. Okay. That is, we are having the ATGC sequence. So by storing the information in as a ATGC sequence, we can use that information for solving the complicated problems, all those things. So in that way also, now the synthetic biologies are very emerging field, and it is having more future prospects. Okay, that is in the pharmaceutical, biopharmaceutical. See, this is one of the example. That is, we use the synthetic biology for synthesizing the artemisinin. This drug can be used for the malaria. So it is obtained from the plant Artemisia. Okay, Artemisia annua L. This plant, through fermentation or through biosynthesis, it synthesizes. This is one of, that is, artemisinin is one of the byproduct of Artemisia annua L, metabolic product. We can call it as metabolic product. So if we artificially synthesize some of the chromosomes, okay, see, we can modify the chromosomes required for synthesizing the artemisic acid, and then incorporate into that particular plant. And then if we allow for the synthesis of that particular one, we can get the overall production of 40% yield, and then in the 95% accuracy. So in this way, it is used for in the pharmaceutical industry. In this way, we can go for the pharmaceutical basis. We are having the switches, oscillators, all those things. Okay, and though the synthetic biology is an upcoming field, it is also having the limitations, because it need to be certified by many regulatory authorities. Okay, so here the startup cost will be very high. Okay, so only it is having many limitations, and we cannot use the synthetic biology for to reverse the economic system. It should not reverse the economic system. So ecological system. It should not reverse the ecological system. So in all these perspectives, this synthetic biology is having many limitations, but it is an emerging field. So we can go many research working workers going on this synthetic biology. So by utilizing this, we can do whatever products we want. So this is what the final conclusion of this top session. So in this session, we had seen about how the genome has to be sequenced, why it is important, and then why it is important for synthetic biology, and how the synthetic biology are very useful in the current point of view. Okay, so that is what we had covered today. So now the session is open for queries. If you have any queries, please ask me. Any queries regarding the technologies, or any queries regarding the topic we covered today? Thank you so much for your patience. Thank you so much for your attending the session. We will end off our session. Thank you for all.